Now, one another supportive class for cell division. Cell division is the formation of new daughter cells from parental cell is called cell division. Formation of new daughter cells from pre-existing parental cells is called cell division. We can also classify the cell division into three groups. These are the amitosis, mitosis and meiosis. First, the amitosis cell division. If in amitosis cell division, the karyokinesis and cytokinesis occur at the same time. The karyokinesis and cytokinesis occur at same time. It's called amitosis cell division. It occurs in the prokaryotic cells. It occurs in the prokaryotic cell. Okay. Then the mitosis. When two daughter cells are produces, when two daughter cells are produces from the single mother cell and the number of chromosome number will same is called mitosis cell division. It occurs in somatic cell. It occurs in somatic cell in our body. And then the another kind of cell division is meiosis cell division. If four daughter cells are produced from a single mother cell and the chromosome number becomes half and chromosome numbers become half is called meiosis cell division. It occurs in gamete mother cell. It occurs in gamete mother cell at the time of gamete formation only. So look at the mitosis cell division. In mitosis cell division, the chromosome number of the daughter cells and parental cell will be same. For that, the mitosis cell division also known as equational division because the chromosome number of daughter and parental cell will same. In case of meiosis cell division, the chromosome number become half in the daughter cell. For that, the meiosis cell division is also called reductional division. Then the cell cycle. Cell cycle is the cyclic changes. Cell cycle is the cyclic changes occurs in between two successive cell division. Means the changes which are occur after the forming a new cells when they get prepared for the cell division. The changes in between two stages, okay, is called cell cycle. The cyclic changes occur in between two successive cell division is called cell cycle. In a cell cycle, we can differentiate a cell cycle into two parts. We can differentiate the cell cycle into two parts. That is interphase. This portion is called interphase, and this portion is called mitotic phase or divisional phase. In interphase, the new shell will prepare for the next cell division. The new shell gets prepared for a new cell division, and this phase means mitotic phase is the divisional phase where a parent cell gets divided to form new daughter cells. We can also separate the interphase into three parts, that is G1 or growth 1 phase, S or synthesis phase, and G2 or growth 2 phase. At first, we come on the G1 phase. Some changes is occur in that phase. Some changes occurs at that phase in a newly formed cell. In that phase, the following changes does occur, which are: first one, the size of cell. The size of a cell will increases. The size of a cell will increases. Next, the cell organelle, the number of cell organelle will double. The number of cell organelle will double, which are present inside a cell. Third one, the enzymes the enzymes means the proteins which are needed for the DNA replication, are produced in this phase. Okay, so RNA synthesis also occur in growth hormone phase, and also protein synthesis occur in this phase. Then S phase. S phase means synthesis phase. DNA synthesis will occur. DNA synthesis or DNA replication will occur in S phase. In that phase, the number of DNA, or the amount of DNA will become double. Okay, by the replication. Replication is the process of forming new DNA from the parental DNA. It's called replication. So the enzymes which are synthesized in G1 phase takes part in replication of DNA at the S phase. Then G2 phase. In G2 phase, most RNA will synthesize in this phase. Most of the RNA will synthesize in G2 phase. Then the concentration of protoplasm will increases. Third, the protein synthesis also occurs in this phase. Then the cell get prepared to divide. Then the next phase means M phase or mitotic phase will occur. Mitotic phase will occur. We can also classify the mitotic phase into two parts, that is karyokinesis and cytokinesis. What is karyokinesis? The division of nucleus. Of a cell is called karyokinesis, and the division of the cytoplasm of a cell is called cytokinesis. The karyokinesis also separated into four phases. The karyokinesis also differentiate into four phases. These are the prophase, metaphase, anaphase, and telophase. In prophase, chromosome will form. In metaphase, all chromosomes are arranged in equatorial plane and attached with the spindle fiber. Okay, attached with the spindle fiber. Then anaphase. In anaphase, by contraction of the spindle fiber, the daughter chromosome will separate. And move to the polar region of a shell. It's called anaphase. And in telophase, new to daughter nucleus will form in a shell. Two daughter nucleus will form inside a cell. At that state, the shell contains two nucleus for a certain period. Then, after completing of the karyokinesis, cytokinesis will occur. Cytokinesis means the division of the cytoplasm of a shell. In that phase, the shell will divide, or the shell separate into two cells. Okay, that is the cytokinesis. The cytokinesis process is different in plant cell and animal cell also. In plant cell, a cell plate will arises by the help of dictyosome or Golgi body. Inside the plant cell. In plant cell, the Golgi bodies or dictyosome helps to forming a cell plate in the middle portion or in the equatorial plane of a plant cell. And then the chemical composition of the shell wall will deposited on the shell plate, and it elongates into two sides. When the cell plate is attached with two sides of the cell, then two daughter cell will produces. Okay, and in case of the animal cell, a furrow will form at the middle portion of the shell. A furrow will form at the middle portion of a shell, and then the furrow elongates inwardly, and the furrow elongates inwardly, and when they come closer or get attached, then two new animal cells will form from a single parent cell. Okay, so that is the cell cycle. Then G0 phase. What is G0 phase? G0 phase or resting phase is a phase is present in G1 phase, or at the starting point of the G1 phase. We also said as the G0 phase is present in between the M phase and G1 phase. In G0 phase, the cell is neither dividing nor preparing for division. Means if any newly forming cells does not divide or does not prepare for next cell division is called G0 phase. It is the quiescent phase. It is the quiescent phase or resting phase of a cell. The G0 phase also classified into two groups: reversible G0 phase and irreversible G0 phase. Irreversible G0 phase means the cells permanently stops its division process. The cells, if a cells permanently stops its division, then it is called irreversible G0 phase. And the reversible G0 phase is the if any cells enters into G0 phase for a certain period, it is called reversible phase, because after that period the cell will divide again. The cell will divide again. It is reversible G0 phase. In case of us, the liver, some liver cells, some cells of our liver is present at reversible G0 phase, and our nerve cell are in irreversible G0 phase. For the completing of a cell cycle, it needed only 1 day. For a completing of a cell cycle, a cell needed only 1 day or 24 hours. If we separate the time period according to cell cycle, we can see the G0 phase. The G0 phase takes 11 hours, S phase takes 8 hours, G2 phase takes 4 hours, and the mitotic phase takes only 1 hour. Means divisional phase or mitotic phase stays or occurs in 1 hour. But most of the time are taken by a cell for preparing itself for the next cell division. So it is the cell division. Then how cell cycle is regulated, and then how a cell cycle will regulate it. So regulation of the cell cycle. In regulation of cell cycle, two special proteins takes part to regulating a cell cycle. These are the one cyclin protein, and another is an enzyme, which is known as the cyclin dependent kinase enzyme. Means the kinase enzyme is which which produces ATP from ADP by addition of an inorganic phosphate. Means ADP plus PI is equal to ATP, and that reaction occurs by the help of kinase enzyme. Here cyclin dependent kinase enzyme means the kinase enzyme is activated at the presence of cyclin. For that, the kinase enzyme is called cyclin dependent kinase enzyme. At the time of regulating a cell cycle, the cyclin dependent kinase enzyme will bind with the cyclin protein by help of ATP or cellular energy, and they form an cell cycle regulating complex, complex, that is the cyclin CDK or cyclin dependent kinase and ATP. Look here. In a cell cycle, three checkpoint will present. Three checkpoint is present in a cell cycle. These are the G1 checkpoint, G2 checkpoint and M checkpoint. G1 checkpoint is present in between the G1 phase and S phase. G1 checkpoint is present in between the G1 and S phase. G2 checkpoint is present in between the G2 phase and the mitotic phase. And the M checkpoint is present in between the M metaphase and anaphase of the mitotic phase. Okay, so how does the work? At the case of or in the case of G1 checkpoint, G1 cyclin is take part. The G1 cyclin is present in that phase, okay, or in that checkpoint, and then the cyclin dependent kinase will bind with the help of a ATP at that point, and they checks the protein synthesis, means the enzyme synthesis which are needed for DNA replication. When the all enzymes which are needed for the DNA replication are prepared by the cell, then the ATP will break down into ADP and an inorganic phosphate, and then G1 cyclin and CDK enzymes or cyclin dependent kinase enzymes will separated, and the checkpoint also removed, and the cell enters into the next phase, means into S phase, and then the G1 cyclin are accumulated, then the G2 cyclin are accumulated at the G2 checkpoint. When the G2 cyclins are accumulated, then the CDK enzyme and ATP also get attached at that point. For that, the cells stops to entering it. The cells stops to entering the mitotic phase. When it will break down, if the DNA replication process is complete, only then the G2 checkpoint will break down. If a cell completely prepared its DNA or completely produces the new DNA, or they're doubling their DNA amount, only at that time the ATP will broke down, and the G2 cyclin and CDK protein will separated, and then the another cyclin protein called M cyclin protein are accumulated at the M checkpoint, and another CDK and the CDK enzymes and ATP is bind with it and form the cell cycle regulating complex or M cyclin complex. It checks all the chromosomes which are present inside a cell will come at the equatorial side of a cell, and the spindle fiber properly attached with the, it confirms the two steps. First step, the arrangement of the all chromosomes which are present inside a cell in equatorial plane, and another is the spindle fiber properly attached with the centromere of each chromosomes. If it does occurs, then the ATP will break down and the M cyclin and CDK gets separated. Then the CDK protein also attached with the G1 cycle in next process, and it going on. Okay, So it is the regulation of the cell cycle.